Hey everybody, so we're going to look at the cell cycle in this lesson. Okay. So the cell cycle is basically the way that cells duplicate or reproduce. So cell division is critical for life. If you have damage to your cells, we need a way to replace them. So it allows for reproduction of cells, the growth of tissues, and tissue repair. So the cell cycle is basically the life of a cell from its formation until the point at which it divides. So when we talk about cell cycle, we're talking about cell life. So to be able to do this process, they have to organize and package DNA before we can divide, because the idea is that you've got to make sure that the cell always maintains the same amount of DNA in every single cell that exists. So before it can divide, it must make copies of that DNA, so we have just enough to make two cells. So DNA associates with and wraps around proteins that are known as histones to form nucleosomes. So think of it this way: if you have a pencil and you wrap a string around it, the pencil would be your histone, the protein, and the string would be your DNA. And once they're wrapped together, we would call that pencil string complex a nucleosome. Strings of nucleosomes. So we've got one pencil string concept attached to another pencil string, attached to another pencil string. And so these all come together to form what we call chromatin. When a cell is not actively dividing, the chromatin is what we call in a non-condensed form. So it's not all wound up so much as it's kind of spread out more. After the DNA has replicated, then it's going to condense to form chromosomes. It's going to get all wrapped up really tight and form those chromosomes, which are those X structures we almost always look at. So they're really densely packed. And the reason why is because it's a whole lot easier to divide, to move them around if they're all coiled up as tight as can be. So since the DNA was replicated, each chromosome has a duplicate copy. So here you see we actually have two separate chromosomes that are attached to each other. So the copies join together and form what we call sister chromatids. So our sister chromatids here, one chromosome, two chromosome, together they're sister chromatids. And they're held together in that middle with a centromere. That basically is that region right there in the center that holds them together. And then we have the kinetochore that are proteins attached to the centromere, and they're going to link the sister chromatid to mitotic spindle fibers. Basically, think of the spindle fibers as we have lassoed, and we have the rope that we can then use to pull or control that individual chromatin. So DNA winds up, wraps around to form our nucleosomes, and then our nucleosomes all pack together to form the chromatin. And then our chromatin in the right times will condense and pack up to form a chromosome. And then our chromosomes will pair up to form our sister chromatids. All of this is happening in the nucleus. So the genome is all of the genetic information. Therefore, we're saying all of the DNA that's found in an individual cell. Prokaryotes are going to have a singular circular DNA. So there's just one kind of ring of DNA, but then they have some other little rings that have like extra stuff, but that's not their key DNA. Eukaryotes are going to have one or more linear chromosomes. All of these that we're looking at in this picture, those are linear. So that's what our X's form out of. Every eukaryote has a specific number of chromosomes that will be found in every member of that species. For humans, that's 46. 23 pairs, total of 46 chromosomes. If there's an extra chromosome, it can lead to issues. For example, Down syndrome is caused by trisomy 21, meaning there's an extra, there's 3 instead of 2, an extra chromosome number 21. And so you can see how going away, not maintaining homeostasis for that specific number of chromosomes can cause problems. Chimps are 48. Elephants have 56. Okay, so a quick practice free response question. Biological structures tend to be composed of smaller units that assemble into more complex structures. Using eukaryotic chromosomes as an example, A, describe the smaller units and their assembly that leads to the larger, more complex structure of a chromosome. And B, identify one major function of chromosomes. So you would want to pause and work on your practice for your response. Okay, so we have 2 key types of cells, and it's important to know about these different types of cells because some of the behaviors will be different based on these general types. So we have somatic cells and we have gametes. Somatic cells are body cells. They are typically diploid 2N, meaning they have 2 sets of chromosomes, one from each parent. So you are made up, all of your, the majority of your cells, the cells that are somatic cells, have a total of 46 chromosomes, 23 pairs. 23 of the chromosomes came from your mom, 23 of the chromosomes came from your dad to make the 46, the diploid number. These cells will divide by mitosis. So humans, 2N is 46, 23 from mom, 23 from dad, and they're going to divide by mitosis. Gametes are reproductive cells, eggs, sperm. They are going to be haploid or N, meaning they only have 1 set of chromosomes. That's how you got 23 from mom and 23 from dad, 23 from mom's gamete, egg, and dad's gamete, sperm. They're going to divide through the process of meiosis, which we go into in more detail in the next unit, but we're trying to get the big picture here. So the idea with meiosis is that we're going to divide and divide. So we divide twice. So in humans, a gamete is only going to have 23 chromosomes rather than the normal 46 that we find in somatic cells. So in this unit, we focus on mitosis, but we touch on meiosis, and we'll go into more detail on meiosis next unit. That's the reason why the lab that we do with the mitosis unit kind of jumps between the 2. Okay, so finally, we're in the cell cycle. And so we're going to go through each of the parts. So the cell cycle has alternating phases of interphase and mitosis. So you'll notice the orange all the way around here, that's interphase, and there's a little teeny tiny section that would be mitosis. G1, S, G2, mitosis, and cytokinesis is kind of how we break it down. G1, S, and G2 are interphase. Interphase is the longest portion. It's about 90% of the time. You would expect, if you were looking at a tissue, to see most of them in interphase, assuming it is a normal, healthy, regularly dividing tissue. G1 is known as the first gap phase, and the cell grows and carries out normal functions. I always think of this as growing from a baby up to just before you hit puberty. So you grow, develop. S phase is synthesis phase. This is where DNA will be replicated and chromosomes are going to be duplicated. This is puberty. This is that point where it's possible to divide to make a baby. But the reality is you're not actually really ready to do that. Just because you can doesn't mean you should. We move into G2 or the second gap phase. This is what we call the final growth and preparation for mitosis. So this is where we're actually taking the time to make sure that we really have everything ready to make a baby cell to divide. You know, like getting a job, a place to live, all of those kind of things before you actually have a baby. And the last is the M phase here. Mitosis, that's where the nucleus divides. This, a lot of people get confused. They're like, oh, mitosis, cell division. But in reality, mitosis is nuclear division. So we're going to take the DNA and the nucleus, split it up and prep it for two separate cells. And then cytokinesis is actually the cytoplasm or cell division itself. So you want to make sure that you are clear that mitosis is nuclear division, cytokinesis is the rest of the cell. At the end of mitosis, we should have two identical diploid daughter cells that basically are clones or exact copies of the original parent cell before we went into the division. Okay, so hopefully that gives you a good idea of what the cell cycle is and how it works. And I'll see you in the next lesson.